make sure that you're doing the lab homework, right? That's due Wednesday at 11, okay? So make sure that you're getting that done. And then I will be opening another homework assignment for lecture on the Bryophytes. So I'll get that situated and I'll let you know. I'll send out an announcement, okay? All right, so we've covered the mosses, which you actually see on this diagram, this phylogenetic tree, that they're the most advanced of the bryophytes when you look at this. So the further it is away from the trunk or the trunk of the tree, so to speak. And so we've covered the mosses, and you can see they're the furthest away of the bryophytes from the ancestral green algae. So now we're going to get into the liverworts and hornworts, which are over here. And some people think that the mosses are actually, well, the way they drew it here, they're implying that the mosses are pretty closely related to this next group over, the lycophytes. Those are the seedless, part of the seedless vascular plants. So there's still some debate, but currently bryophytes, the mosses do have quite a bit of characteristics that they share, but they are studying the mosses a little bit more carefully and asking these types of questions. So that's one thing in biology. The classification scheme tends to shift over time as we get more data. All right, so here you're looking at a picture of two different styles or types of liverworts. So I think you saw in lab last week more like what the one on the right looks like. That's the phallus type liverwort. So Marchantia is a good example of that. And then the one on the left is a leafy version. So of the liverworts, they can really be broken down into those two morphologies. They're either leafy or phallus. And actually, there's more leafy liverworts out there by far. All right, so leafy liverworts win. However, like I tried to buy some leafy liverworts, fresh leafy liverworts for lab this week, and they didn't have any in stock. And so they tend to carry the phallus kind because Marchantia is very famous for some extra fun things to look at. So we tend to focus on the phallus, but it's actually the leafy. When you count them all up, all the different species, the leafy win. And if you looked at the leafy liverwort, you might say, geez, that looks like a moss. All right. And so one of the things that I do when I'm out hiking around and I'm like, is that a moss or is that a liverwort? One thing I'll do is I'll look very carefully and I ask myself, is it radially symmetrical type morphology or bilaterally symmetrical? And liverworts fit into the bilateral where mosses are radial. So we're bilateral. There's only one way to cut yourself in half to have a mirror image, okay? And it's the same with the leafy liverwort. If you go back to that picture, there's really only one way to cut those liverworts in half to have mirror images, okay? So that has saved me many times when I'm trying to figure out if that moss or liverwort. And so leafy liverworts in general are pretty happy in really wet areas, more so than mosses are. And that's another thing to think about too. And so if you really want to see a lot of these, go south, go into the tropical jungles. And another characteristic is the leafy liverwort have two partially overlapping leaves. They're not true leaves, but that's why I put them in that little quotes there. But that's another thing to look at when you're studying the leafy liverworts. But so if you're in the tropical areas, you're going to see a ton of leafy liverworts and very few of phallus kind. So phallus is only about 20%. But like I said, in botany classes, we tend to focus on some of the phallus because they have some additional fun things that we can look at. So phallus type, they're flat, they look like ribbons, and the classic example is from the genus Marchantia. And one of the reasons that they sell this one all the time is that it goes through asexual reproduction a lot. And you can see it doing that visually because it'll produce these so-called gemma cups or splash cups. And so if you see those, you know that they're kicking into asexual reproduction. So there are these little cups on the top of the thallus body. And if you are lucky enough, you'll see little tiny pieces, look lens-shaped or ovoid-shaped pieces of tissue sitting in that cup. And they're attached very delicately on one end. And so what you can imagine, I'll show you some pictures, but basically they call them splash cups because if rainwater dives into that cup just right, it splashes out that tissue very easily. And hopefully it'll bounce out of there, go somewhere close by, suitable, and start to developing. So leafy versus thallus. And if you put it here, they're showing Marchantia's life cycle. And again, you know, they're trying to show that the gametophyte phase is up top. So that's usually how we are thinking about dominating when you draw it out like this. And so you can see there's a male and a female gametophyte. A couple of extra terminology on here I would like you to be familiar with. So they do some additional really fun structures that are fairly unique. And so with the male gametophyte on their thallus body, they'll shoot up these structures that are called antheridophores up top there. And so anytime you see P-H-O-R-E in a word in science, it means to hold up or to bear. And so what this is implying that it's just this special stuff that will hold up the antheridium. All right. And if they kind of look like funky umbrellas. They remind me of little umbrellas. And so here they're doing an enlargement of that. And so at the very top of this so-called umbrella portion, that's where you have all the antheridia, lots and lots and lots of sperm and antheridium. And they're trying to show a raindrop with a lot of sperm getting splashed. Okay? And then if you look at the female gametophyte, which is hopefully close by, nearby, so that the sperm can easily get there, they too have a structure, a stalk that is holding up or bearing. So we call that the archegonia form. So holding up or bearing up the archegonia. And so again, they're enlarging it right here, and they're showing, to me, the archegonia for kind of looks like an umbrella that's been through a hurricane. It's all tattered and kind of beat up. It's not this flat. And so, and then the other thing too is that the archegonia are tucked underneath. They're not on the surface, which to me is like, how the heck does a sperm get there? But it does. But you got to get the splashing just right in order for the sperm to splash up into the archegonia. And remember now, each archegonia only has one egg, and then hopefully fertilization can occur. Yeah, I think that is a mechanism where they're trying to really tuck away their precious eggs because there's only one in each archegonium. And so I think it is a protective mechanism because if you don't have, you know, good healthy eggs, you're toast. This isn't going to happen, you know. And so with sperm, you know, there's massive amounts of sperm. So if some don't make it, it's not such a big deal, you know. They're playing the odds on that. But again, they're trying to show you need that splashing of water. Yeah, it's pretty amazing. You usually see the male and the female together in a cluster, but still, it's challenging to think of that you got to get that just right. But if that does occur, then that egg does it gets fertilized, and so then we call it the zygote. Okay, so we're back into the diploid state after fertilization. But remember, that egg doesn't go anywhere; it just sits there and starts to grow. and grow and divide and divide. So it just goes through rounds of mitosis, and it's growing until it finally becomes a mature sporophyte. Now, you can't really see the sporophyte too well. And I'll show you some actual pictures here, but so here they're showing on the far left the Archegonia 4, and you can see these little brownish things sticking down, like from the top of the tattered umbrella. These little things are the sporophytes. They're quite small, and you can see, they are a little bit, they have a little bit of a stalk. And then they have a sporangium, and that makes sense that they still dedicate a spot for spore production inside the sporangium. And then they have to go through meiosis in order to get back into the haploid gametophyte phase. And then they're trying to show these little brown pieces of tissue. They're called elaters. Okay, they're trying to point to that. So these pieces of tissue are kind of dispersed in the sporangium. They're kind of long and skinny. And they go through, when they dry out, they kind of twist. And so as the sporangium tissue cracks open, the elaters will start to twist. And then that kind of causes those spores they think it helps with spore dispersal. So it's a little extra adaptation to try to get those spores dispersed nicely. So they have these antheridiaphore and archegoniophores that kind of hold things up off of the rest of the gametophyte. But on the other hand, their sporophyte is quite small, because it's hanging down right where the egg got fertilized. It grows right there, and it doesn't get very big. So let me show you. I'm going to jump ahead a little bit. I'll come back to those other pictures. So here is the left picture is the antheridiaphore structure. So you see the gametophyte. The top of the umbrella, so to speak. And then on the right, you can see the more scattered look to the part. So visually, once they're mature, you can tell them apart just by that difference. And you can tell the male versus female. And so those are nice, like side-on shots. Here's, if you were looking down on it, this is some antheridiaphores. So sometimes it's hard to spot. You have to really look carefully. Here's a really nice picture of some antheridiaphores. And so it's in the top of the umbrella that you have all the antheridia, and all of that dark blue. Those are all, it's just a ton of sperm in each one. And then here you see the archegoniophore and antheridiaphore. So you see how they're clustered here. And so the more tattered ones are they can spot that even in the back of the room, that tannish structure. So that's the sporophyte peeking out. So that egg right there. That is growing down. Here's another image where a lot of them got fertilized. And you can see that whitish structure kind of peeking out. That's the sporophyte. So very small, very petite and dainty. Here's a really nice shot. So the sporophytes are hard to spot. You really have to look carefully. If you look at that underneath, or the antheridiaphore, this is an archegoniophore, and then this is a fertilized archegoniophore. So now it's formed the sporophyte. So that's what the sporophyte looks like under the microscope. So you can see that egg got fertilized, and then they moved it sporophyte in the soft area. Most of it is sporangium that you have the spores being produced. So it's really petite and kind of tucked under there. go back up. So it's the Thallus type, or Chantia, in this case, that we're looking at, that will do that kind of funky archegoniophore and antheridophore. But if they're going to go through asexual, then they form those gemma cups. And that's what you're seeing here as you look down on the gametophyte. Lots and lots of gemma cups. And then, like this one, you can see there's a little bit of tissue in there. Here's a close-up. And if you look really carefully, you can see some tissue pieces in that one pretty nicely. So if you see those structures, you know they're going through asexual reproduction. And if you did a microscope prep of that, it would look something like that, where you're looking on side of the gemma cup, and all the purple are the little bits of tissue, and you can see how delicately they're attached. So you can imagine, if you think of a big, water drop coming in there, it's going to splash it right out. And hopefully that tissue will land somewhere close by and it'll grow into the gametophyte. So liverworts are really quite good at hanging out in really wet environments, consistently wet, and they can do fine with that. There's even some aquatic ones that you might be familiar with if you have aquaria. So if you've ever gone to a pet store or whatever and bought some plants for the aquaria, this is Reishi, and it's an aquatic liverwort. And a lot of people like to put that in their aquaria because it does provide a lot of oxygen. All those bubbles that you're seeing, that's, nice oxygen production. So that's actually where a lot of people, whether they knew it or not, they have been working with liverworts, aquatic liverworts. So aquatic liverworts are out there too. This is just a really nice picture showing these air sacs that they can produce in order to stay buoyant. And other organisms like rotifers like to hang out in there. So, that reminds me. This Thursday, hang on, I'm going to stop share for a sec and pull that up. Where is that? This Thursday, there's a seminar. I was going to offer extra credit seminar if you can make it, because it's one of our grad students, Emily Midlowski, who is giving a talk. If I can pull that up. Where is it? Liverworts and how do I do this? Project. Oh, that's not what I wanted. All right. Oh, Emily is riding a rotifer apparently in this image. So she's giving her talk this Thursday at 1 o'clock in this building, one of the big lecture halls. Not the main one, but one of the ones on the, I guess, on the right. So if you would like to go, and for extra credit, if you can attend and take some notes, so I know you were awake, and hand that in. Oh, that's right, Thursday. Yeah. No, unless they record it. I don't know if it'll be recorded. I could ask. So your lab starts at now. As I say, if it started at noon, you might be able to, but no. I'll see if it's recorded. So hold off on that idea. If it's recorded, then it would be fair because everyone can watch it. Gave a talk. It was like very close to being. Yeah, okay. Well, I think what I'll do is, I know Emily quite well. I'll ask her if she'd be okay if we recorded it. And I think, yeah, because she used to teach this lab with me. So she's like all over the Bryophytes. She loves them. So, obviously. So I think if I told her it was, for this class, she'd be like, oh, yeah. So hold off on that thought. But I thought that when I saw that that was coming up, I'll be certainly going to it. But okay, so let me go back to sharing. She just got accepted. She's going to do her PhD up at Michigan Tech University. Very, very happy for her. It's going to be in the Department of Forest Science. And I think she'll be working on Isle Royal. So she's very happy for her. She'll love it up there. She's the type of person who will embrace the Keweenaw. So, okay. So liverworts, really, really interesting. The last group I wanted to talk about are the hornworts, which we don't really have many up here, as far as I know. Anthoceros species is one of the few temperate ones. Most of them are tropical for sure. There's not many out there that are still alive, haven't gone extinct, only about 100 species. They kind of look like Marchantia, the thallus liverwort type, but they're more rounded. They're not quite as ribbon-like. They're more of a roundish shape. And you really have to look hard to find any Archegonian antheridia because they're really low, like actually in the gametophyte thallus. They're not held up like we saw with Marchantia. And they often have pores in the gametophyte where cyanobacteria hang out, especially nostoc. And nostoc likes to hang out with a lot of plants in general, but especially some of the bryophytes. And nostoc is very, very good for the plant to have around because nostoc can fix nitrogen. So it can grab dinitrogen gas from the atmosphere and convert it into a form that the plant can readily then use. And so nostoc will do that and share some of its fixed nitrogen for the hornwort. So if you're a hornwort, that's a really good relationship to be in. And then the cyanobacteria does get some growth factors and nutrition from the hornwort. They're called hornworts because when they shoot up their sporophyte, it's very obvious. It's just these long horns, essentially long and straight. And they actually, once they form those, they actually have meristematic tissue at the base. And so it can actually persist much longer than what we see like with the moss sporophyte. That, you can usually see the moss sporophyte too, but that one's a little bit more ephemeral compared to the hornwort. And so it's green. They've documented that it can carry out photosynthesis. So it's a little unusual in this regard because, usually we say that the bryophytes, the sporophyte is dependent on the gametophyte. And so they are showing, yes, it can do photosynthesis, but I'm not sure that it could get all of its needs met with the level of photosynthesis that it does. And then it too also has those pieces of tissue called elaters that will twist and allow for spore dispersal. And then so it's this long spike or horn. And then when it's got the spores to go through meiosis and everything's good to go, then it just splits all the way down from tip to base. Here they're trying to show that in this diagram where it's starting to split open. So it kind of gets split in half lengthwise. And then those two parts peel away from each other and the spores, hopefully with the help of the elaters, will release. Yeah. Yeah, there used to be more, and now there's, and I don't know exactly why the hornwort was having a harder time, but yeah, compared to like mosses, mosses are thousands of species. So there's just not a whole lot with the hornworts. But if you do happen to see one, they're really fun to look at, especially when they have the sporophyte. This is, and we would never ask you to identify this visually because it's really hard to see, but these are antheridia that are in the gametophyte, those little roundish structures, those are antheridia. There's a close-up of one. So very, very small archegonia, and again, we wouldn't ask you to identify this, but just trying to show you, I mean, this is a super magnification to be able to see this. So, but if that egg in the archegonium gets fertilized, then you would start to see this kind of growth where you have the sporophyte starting to grow right where the egg was, because egg was fertilized. So now it's going to become the sporophyte. Here's a drawing of that, just trying to show that there is meristematic tissue at the base and that you have this elongation, the later tissue, spores. They even show that it has stomata. So it is long-lived compared to what you see with the other two groups with their sporophytes. So here you can see it's nicely green, the sporophyte sticking up off the body of the bryophyte. And then if you were to take that horn and look at it, this is just a partial image of it under the microscope. So all the reddish, those are all the spores that are formed. So here they are trying, this is a study where they were looking for nostoc in the underside of the tissue there. And they were finding little tiny like crevices or depressions where they could find a bunch of cyanobacteria hanging out, nostoc. And then here is another one where this is Anabina in this case. Anabina filamentous type cyanobacteria. So it's kind of hard to see, and I wouldn't have to big on, but this is there's several in little like depressions. And so again, if you're that hornwort, it's really good strategy to hang out with these nitrogen fixing cyanobacteria. Okay, the last slide, I would like you to make sure that you start the process of memorizing the phylum names, please. So moving forward, we're going to be doing that a lot. Different groups of plants. So you only need to know 3 for the bryophyte and 3 phylums. And certainly we're not going to get too far into that hard plants considering the vastness, but for these 3, you can do that. So phylum Hepatophyta, so hepata is referring to liver. So if you can make that connection, we're talking about the liverworts. Anthocerophyta, the way I remember that is antho and antler isn't too far away from each other. So I would think about hornworts. And then the Bryophyta, I don't know, I didn't come up with a saying for that one. But this is kind of a nice summary table showing you the difference in just population levels. You can see that the mosses win. And then some of the main characteristics of the gametophyte versus the sporophyte and their habitat. So this is kind of a nice one-stop shopping for just trying to get the 3 different phyla down, some of their characteristics. Okay. Any other questions on the bryophytes? Cute little things. So keep your eye open for them in the spring and through the summer. They're all over. If you look carefully, they're there. And remember, they don't have true roots, so they can hang out in areas that is very devoid of soil. Rocks and cement and gravestones, they're all over because they don't have to, find some soil to put their roots into. They don't have roots. So in that way, they don't have to compete with all of the vascular plants. Okay, I'm going to stop share and get into the next slide set. I think that should be in EDUCAT, the seedless vascular plants. I'm SVP. back to. Okay, so the next group of plants, which I think you'll be looking at in lab this week, is seedless vascular plants. And so now we're shifting into a very big transition because they're getting vascular tissue, but no seeds yet. And so it's an interesting kind of transition here where bryophytes had no seeds. All we talked about were spores. So there was no seeds there either. And they didn't have vascular tissue. Now we're shifting into a group of plants that developed vascular tissue. They evolved that mechanism, but they still didn't quite get there with the idea of a seed. All right. And so, and when you look at this group, their vascular tissue is the more primitive type of vascular tissue compared to what we were looking at and focusing on with the angiosperms. So usually, when you think about the seedless vascular plants, this is a huge fern that you're looking at here, and they're an example of a seedless vascular plant. And that's probably what most of you are most familiar with. But when you look at the seedless vascular plants, there's 4 main phyla. And so the top left, those are the club mosses, which aren't mosses, remember, so be careful about that. And then the top right is a funky group called the whisk ferns, because they kind of look like a whisk broom when you look at them in a cluster. And the bottom left, those are horsetails or equisetum. And then the bottom right is an example of a fern. So 4 different groups. And when you think about their evolution, they're trying to show here kind of a nice distinction on this left one where you can see, the non-vascular plants we just covered, the mosses, liverworts, and hornworts, and then you can see the rest of this diagram is all vascular. All right, but then you divide it further into, okay, yeah, they're vascular, but then the other classification is, do they have seeds or don't they? And here you can see that you have those options. All right, so we're going to get into the seedless vascular plants. The one on the right here is also showing how you have the seedless vascular plants. And then you can see the gymnos and the angioles diverging off those. And then if you go to this one, which we've seen, so now we're getting into the seedless vascular plants, which are right here. So 4 phyla. Okay. So these are extinct ones. This is only from the fossil record that these drawings were taken from. And so this is just showing some of the ones that were here, but have since gone extinct. And we're going to see that there are some of the seedless vascular plants that very much look like this, like the whisk fern looks very much like the rhinophytes. And then club mosses aren't too far in the zostrophill. So they can track it in the fossil record. So the common names here, horsetails, whisk fern, club mosses, and true ferns. So they say true ferns versus a whisk fern. That's why they name it like that. So this vascular tissue showing up now, this is a huge evolutionary leap, getting this to happen. It really did allow for plants to get much bigger and to get into much drier habitats because they could rely on the reservoir of water in the soil. And so even if there's periods of drought, as long as there's some water in the soil, and remember, roots can go quite deep, then you can have a success with these vascular tissue. So big shift here and allow them to be successful in drier habitats. We still are going to see that there's going to be an alternation of generations going on, but now we're going to flip it It's no longer the gametophyte that's dominant, it's the sporophyte. So from now on, it's the sporophyte that's dominant through all the rest of the groups of the plant. So that's a big switch. And so when we say it's dominant, that means that when you're out hiking around, that's what you see. It's the longest lived portion of the life cycle. And when the gametophyte develops with this group, sometimes you can't even see them because they're underground. So you might not even, I mean, it's very easy to forget that they have an alternation of generations because often you just don't see it. So it's smaller, the gametophyte's smaller, and they tend to break free from each other pretty quickly. And I'll get into that a little bit more. So they say they're nutritionally independent because they don't stay connected through their life cycle. They don't do that. So we still see some of the same patterns of development, though. We have antheridia, where you have tons of sperm showing up in each antheridium, and they're motile. And then you have a single egg in each archegonia. And so that's all the same as what we've just talked about with bryophytes. And it's also the same that they have to have free water in order to get the sperm to be. But this shift into the sporophyte phase is dominating. So with the seedless vascular plants, we tend to write the alternation, we draw it this way, where the sporophyte's on top, just to try to, again, really get at the idea that that's the dominant phase. But it's the same process. We haven't changed any of that terminology. Which phase is the one that's dominating in that plant's life. And so if you're out hiking around and you see, a fern frond, that is the sporophyte. It's nutritionally independent. So soon after that egg gets fertilized and you get the zygote, you tend to see this idea of nutritional independence. They're not, the sporophyte isn't depending on the gametophyte. And then, like with ferns, you can have perennial sporophytes that will keep popping up new fern fronds every spring, and they're just perennial. They just keep coming back and coming back. Ferns are great for doing that, and so that's just all asexual. Right? That's just the sporophyte re-sprouting every spring. And if you do have ferns, especially like bracken ferns, if you have any of those on your property, they can be very challenging to get rid of. That's what you're trying to do because those rhizomes are underground and every spring they keep shooting out new fern fronds. Yeah, I see what you mean. Yeah, I mean that's a fair way to think about it. Yeah. Mm-hmm. So ferns can be quite beautiful. I had one where I was renting one time. They allowed us to landscape a little bit, but the thing with that is you have to remember when they die off every fall. The fern fronds will die off, so you have to. If you're going to landscape, you have to be thoughtful about where you're going to put your ferns. You have to have other plants around. But anyways, gametophyte, very short-lived, typically, not always, but typically. And then they're very small. Sometimes you can't even see them with the naked eye. And again, they kind of separate from each other. And that tends to be it's not 100%, but that's more often than not. And some are actually what we say saprophytic, which just means that because especially if they're underground, they can't really do photosynthesis. So they are saprophytic. They'll absorb nutrients from decaying organic matter around them. There's some interesting exceptions with horsetails and most of the ferns, their gametophyte are green, and you'll see that they're doing photosynthesis, but they're tiny still most of the time. And they actually might mistake them if you had your nose close to the soil, looking around. You might go, "Oh, look at that Thallus liverwort," but in actuality, it might be a fern gametophyte, for example. So, and then sometimes the gametophyte will go ahead and reproduce asexually. They'll just fragment and be able to grow into a brand new gametophyte. So, the gametophyte and sporophyte, they can, they're pretty much independent of each other pretty quickly. and they both can do asexual. But again, the terminology that you're learning is going to stick. All right. So we have the archegonia producing one egg in each one, antheridium and each sperm. And then the sporangium tissue on the sporophyte, it's producing spores. Now here's a new term that I want you to know, strobili, because that's going to keep showing up again as we move forward into the other groups of plants. So some of the seedless vascular plants, what they do is they group their sporangium tissue together very tightly. And so it taken as a whole, you can see this nice big cluster or aggregation of sporangium tissue. And so then we start referring to that structure as a strobilus or strobili for plural. And it's usually at the tip of stems that you'll see this. And there's a little different designs, but when you look at it, you can understand why they think that it's probably the forerunner of the flower. And so that's why I want you to know this term, strobulus, and be able to identify it on the seedless vascular plant. Okay, so some of the same terminology, but then the strobulus idea is being shown here. So this is on the left, you have a club moss, and you can see the darker green are where the bulk of the photosynthesis is occurring, but then you see the lighter green, which my thing won't show up on there because it's red background, but the lighter green, you can see how much more tightly packed is. So those are strobili. And then on the right, these are really funky looking, the horsetail plant and stem plant. So each one of those knobby looking are strobili. I mean, sporangium, sorry, sporangium. And then so it's all clustered together to create. So you would say, oh, look at those 4 strobili in that image. Okay. So the seedless vascular plants, when you look at them, they like the other terrestrial plants, they have chlorophyll A, chlorophyll B to do photosynthesis. They have carotenoids to help out with photosynthesis, give some different colors. If they have extra sugar to go around, they'll pack it away and store it up as starch. They have cellulose in their cell wall, just like what we've been talking about when we went into the plant anatomy idea. Now, the vascular tissue that they do have, goes xylem and phloem. With the xylem, remember, those cells are dead at maturity. And back during the plant anatomy section, we said that there's 2 types of xylem, tracheids and vessels. Now, with the seedless vascular plants, because they're more ancient, they only have tracheids. The vessels aren't showing up yet. Okay, they don't show up for a few million years. So tracheids are what they have. That's their version of their attempt at vascular tissue. Phloem, remember, phloem is living cells. They're highly modified, but alive. And when we were learning about phloem back in the day, we said sieve tube elements and sieve cells were the 2 types. Well, these plants, they only have more primitive sieve cells. Right? So sieve tube elements are showing up. And I need you to understand that. That's a big evolutionary process here that we see, where they came up with this more, it's effective but primitive form of vascular tissue. Right? Yes. So vessels were born. No, tracheids are for water movement too, but they're definitely more slender in diameter, and vessels are much wider in diameter. So vessels are more efficient at water flow through them. And tracheids are good for water flow, but they're just not as efficient. And that's actually one of the reasons why they think angiosperms took off on the face of the earth, is they figured out vessels, and that gave them the competitive edge. And that's one of the reasons. That's not the only reason, but they think a lot of plant physiologists point to the vessels And that was a huge strategy that really gave them a leg up in dominating. So going back to the plant anatomy idea. So the sieve tube elements show up in the angiosperms, and yes, they'll have the companion cells. The sieve cells, on the other hand, they're just all by themselves. So sieve cells are a different beast compared to the sieve tube element. It's different. And in fact, we know relatively nothing about sieve cells compared to sieve tube elements. Unfortunately, the angiosperm, they get all the attention with the sieve tube elements. Sieve cells, they just seem to live their own solitary life. They don't seem to have a helper companion cell, at least as far as we know. But again, the research is lacking. So the sieve cell, yeah. When we say a sieve cell, it's the longer. Yeah, so well, the sieve cells, what they do is they connect to each other. So they're like long and skinny, and then there's another one on top, and then another one, and another one, and it forms, taken together, it's a conduit system through the whole plant. And then the vessels, I don't know if this is going to show, well, kind of. The vessels, they're the ones that we always talk about. It plates on head, it plates, but that's vessel. You don't see that. So the tracheids are long and skinny. Vessels are a little bit shorter and fatter, but more efficient. But the seedless vascular plants do not have vessels. Okay. All right, we're going to get a little bit more into vascular tissue next time. And then in lab this week, you'll be looking at different types of seedless vascular plants.